Hello. I think I'm clearly audible. Hi. I think I'm clearly audible and visible. Before I start, I just need to be very sure. Very good evening, guys. How are you? Okay, so just give me a minute. I need to be sure because, yeah, it's showing live on the channel now. Yeah, it's going fine now, confirmed. Okay, so wish you very good evening, good afternoon, good morning, whatever time zone you are in, and accordingly we will be moving. So, guys, my name is Anjali Ahuja. Though you have seen me on the channel, our YouTube channel, so many times, I'm biology teacher plus HOD here for the bio department in Ask ITians. And today, because your NEET exam just happened on Sunday, and you definitely would be looking for the solutions, and you must have got solutions also. But that's not the point. We need to understand the question and we need to get into the depth because now you people will be waiting for the final solution that NTA is going to release. And after that, you can challenge the answer key also, right? So we need to discuss that. If you feel that answer is wrong or you want to challenge it, you can challenge it here also. And for that, you need to be here live with us. And let's start with the session. Let's start with the session. So guys, FYI, we are going to keep 7 to 8 webinars for physics, chemistry, and bio. There are 200 questions in one webinar. We will be discussing 25 questions. So for bio, it is the first session. I will be taking 3 more sessions also. And for physics and chemistry, total 4 sessions will be there, 2 to matlab, and 25 will be done in one question. And as you do already know in the, what do we call, webinar introduction also, it's mentioned that what paper code we are going to solve. So let me share my screen and let me show you what we are going to do today. And also, I'm feeling so happy taking this webinar because we recently got our 12th class result also, 10th class result also. And we, from the morning, we have been just collecting data and all the amazing results people are getting. So the team is really happy. That result will be shared with you also soon. And let's start with the session. Okay, I think I have started sharing my other YouTube link where I was checking myself whether everything is okay or not, but now it is fine. Okay, so guys, now let's start with a webinar. As you do know that our NEET UG exam, it happened on 7th of May. It happened from 2 to 5 shift in India. And there were 200 questions, all the analysis, how the question paper happened, what type of paper question were there, segregation of 11th and 12th, everything I've taken in the last webinar that you were watching. Today, we will directly start solving the question paper. As you do know, paper code is F6. Guys, let me put a reminder to you on that part that your paper, it had a total E1 to E6 paper code, then F1 to F6 paper code, and G1 to G6. That makes total 18 paper code almost, but question was same. All the question were same in all the question papers. So don't feel that, if it is not your paper code or something like that. All questions are same. It's just that sequence were different. So you definitely, if each and every student is going to take benefit from this webinar. So let's start the session. Okay, so physics session, that physics teacher, I think Dipesh sir will be taking it soon. Let's jump to the bio section. Okay, nothing. I uploaded it with the solution. I didn't realize, but that's okay. I'll hide it because you people are in the chat and you're gonna reply me. I'll give it some time, though you have solved it, so not an issue. Okay, so guys, let's start solving the paper. I have to move gradually. Okay, you can see the answer, but that's okay. So for maximum question, I'll try that you don't see the answer, but would drag me a little, minute things happen. So anyways, movement and accumulation of metal ion across a membrane against their concentration gradient can be explained by. Guys, you people, I'm pretty sure a ninth class student can solve the question, because there's a topic called transport across the membrane. And we do know that there are two types of transport, active and passive transport. Active is the one where ATP energy is needed. Why? Because molecules are moving from low to high concentration. Passive may molecule move from high to low concentration. Low to high, did I say? Yeah, I said low to high. Na? And their molecule are moving from high to low concentration, and no ATP is needed. Now, guys, if a plant needs energy, if a plant suppose need any mineral, if it need mineral, it need mineral. And that's very important, because minerals are important for the plant. Imagine a plant need calcium, manganese. Even if it is less in the soil, it will use the power, it will use the energy and take it in the plant. You're getting my point? So minerals, even amino acid, glucose also inside the plant. But if we talk in the roots, minerals move by active transport. Even though that particular salt mineral is present in less amount in the soil, energy will be utilized. So definitely third is the answer. Let's move ahead. Let's do the next question. Among the evil quartet, which one is considered as the most important cause driving extinction of the species? This question is so easy. I mean, I don't know whether you read it properly or not. Let me show you something. I'm going to show you a picture. You remember you have done this biodiversity chapter, and there you must have done this topic. Extinction. Okay, this picture has a black background, but I think you can understand it. Evil quartet means that these are evil and they are present in 4 in count, and they all are causing the loss of biodiversity. That include habitat loss and fragmentation, alien species invasion, co-extinction, and overexploitation. Now let's read the question again, what it is asking. That all the options are given. What they are asking, that which is considered as to be the most important cause. All are causes of it, but you have to tell which is the most important cause. So think about it. Jai Hind. Jai Hind. Okay. Hi. If you can try answering the question, you can try. Come on, tell question, answer for this one. So we are talking about the quartet, because in the biodiversity chapter, you do know that there are these 4 things important which can cause a problem. Okay, your time is up though. It is actually habitat loss and fragmentation. This is the correct. These 4, all of the 4 are creating problems, but this is the one which is the more problematic, which is the most important cause driving extinction of the species. Habitat loss. If species is not having the habitat, fragmentation means that the environment is changing because of the global warming. The glaciers are melting, or there can be any fragmentation in terms of glacier. Is just one I'm talking about. It can be desert also. That the animal is living in a green area, tropical area. Now it is getting converted into desert. There can be any reason, right? So yeah, 4th is the answer. Now let's talk about the next question. Yeah. Identify the pair of heterosporous pteridophyte. You know, in my class, I say this question for thousands of the time, and I always mention this is the most important one. This question is so old that I've seen this question in NEET exam for like 40 to 50 times. I have been seeing this question in the NEET AI PMT whatever paper for last 30, 40 years. Because I'm not giving that paper, I'm watching that PYQs question, so I know this. So all of the pteridophyte, actually, they are homosporous except 2, which is Selaginella and Salvi. These are the only 2 categories which are heterosporous. Rest, all of them are homosporous, just remember. Heterosporous, homosporous ka definition you remember, right? Megaspore, microspore, strobili, cone, I mean. How do we find it? Anyways, so this is the answer. Now let's talk about the next question. If you guys find any doubt, please put it in the chat, okay? I'm trying to hide the answers. I didn't realize I had to upload the question paper, but that's okay. Frequency of recombination between gene pair of the same chromosome. Imagine this is a chromosome and there are gene 1, 2, 3, 4. These are present on it. And these gene pair, suppose they are also 1, 2, 3, 4. So frequency of the recombination between gene pair on the same chromosome as a measure of the distance, that in between the gene, what is the distance? Whenever we hear about this topic, we always come up with the name of Morgan. But that is not the answer, because Morgan was working over the recombination in Drosophila. And that also he was talking about the sex genes. He was studying those chromosomes. He was studying recombination on sex chromosomes in Drosophila melanogaster. So Morgan was not the answer, because many people think about it. I'm pretty sure some of the students clicked on this, but that's not the answer. It is Alfred Stewart Ward, and I have taught this concept in the class, and you should remember this measuring thing idea came from him, that Morgan further explained in the Drosophila. Then he did a lot of research and everything. So now, guys, next question. What is the function of tassel in the cone cob? Cone cob, you know that, right? You eat corn, right? Butta. So then if you see, there is a tassel thing coming out of that cone. And that's not only present in the cone fruit that we are eating, it is also present in the initial stages, when the cone kernels are not there. So what is the function? Come on, try to answer. So answer is to trap pollen grains, because fertilization will happen, because cone cob is actually not fertilized. It is going to wait for the pollen grain, and then fertilization will happen. And then whatever we are eating, that's the resultant of that fertilization. So yeah, it's not helping with the pollen grain. Achha, let me see if I get to upload the diagram. Just a second. Give me 30 seconds. I'm just looking for a nicer picture to show you. If I don't get it, I will not. Then we'll go ahead. Tassels are there. Wait, what was the question? What is the function of tassel in the cone curve? Okay, I found out. I was expecting a better picture, but that's okay. Look at this diagram. The tassels are there helping with the pollen, and silks are also those structure which will later on grow to form the same structure. But anyways, guys, I think you understood the idea. This is how it is helping. Let's move forward. So next is identify. Wait, I could not hide the answer, but that's okay. Identify the correct statement. Detrivores perform fragmentation. Detrivores perform fragmentation. Is that a clear statement? Is that a correct statement? Next is the humus is further degraded by some microbes. The humus is further degraded by some microbes during mineralization. Guys, you do remember you have talked about the process of mineralization, how it happened, what are the steps and everything in your ecology portion. That question is from there. So first is detrivore perform fragmentation. Yes, it does. It does, right? It's not that fragmentation. It's about cutting the molecules into the smaller one. Then water-soluble inorganic nutrient go down into the soil, that we call it as leaching. And after that, they will be accumulated, right? Next is the detritivorous food chain begin with the living organism. No, the detritus food chain start with the living organism. Oh, sorry, it's not. I take my words back. Start with dead organism, because they are detritivorous. They are saprotroph. They need to be dead and decaying material for surviving. So that is what they look for. So this is not a right statement. Earthworm break down detritus. No, they don't take detritus into smaller. Even they whatever organic material, even soil particle, they are taking in the intestine, that is broken down into smaller particles. Catabolic process can be same, but no, this is not the correct statement. It break down the detritus into smaller particle. No, it simplify the soil. It is not only about the detritus. So correct, that makes this ABC only as a correct statement. Okay, now guys, let's talk about the next question. Let me hide answer. I think I did. Wait, given below are the two statements. One is assertion and one is... Acha, theek hai. Okay, fine. I'm not going to show. There are options also. Assertion is Late wood has fewer xylem element with narrow vessels. And you remember into the anatomy chapter, you people have learned early wood and late wood, and this is related to season, summer season and winter season, that how the wood is thicker and thinner, or how xylem is active and more active or less active. So this statement is related to that. Late wood has, actually that is true, because late is getting formed in the into the winter season. Into the summer season, temperature is increasing, so more of the xylem movement happen because they absorb more of the water from the soil, right? Into the summer season, the rate of transpiration is less, and there's so many factors, right? Stomatas are not open throughout the day because that late, what do we call that, long day plant don't get enough sunlight for the longer time that they need. So there are many changes that happen. So definitely xylem element, it must not be there in the late wood. It will be there in the early wood. Cambium is less active in winter. That is what indirectly I told. Guys, I have told this thing earlier also. If you people are solving assertion and reason question, you should be adding the sentence because. So late wood has fewer xylem element because cambium is less active. No, the both sentences are correct, but definitely. Acha guys, I have told you answer already that the assertion reason are true, but reason is not the correct explanation. But just a reminder, I uploaded this picture to show you this late wood and early wood. Look, that the early wood is having more of the xylem element, and into the late wood, how it seems to be condensed. There's another table also I uploaded here. I think it is there. Just where did it go? I did upload a table. Wait a second. Okay, guys, so I'm uploading this picture, but there was a picture, but that's okay. Let's see these options first about the early and late wood. So as you see, early wood is the spring wood, which is in the spring season, and late wood is the late season, where you may say autumn season, but after that. So early wood may cambium activity is more, which is lower in the late wood. So that's the reason that in early wood, xylem element are produced in large number while there are fewer in number. And that is the thing you wanted. Cambium is an altogether term that we use for xylem and phloem, and that is the only thing that you have to understand. So our our problem is solved. So I think I can proceed to the next question. So already they have written both assertion, reason are true, and reason is the correct explanation. Next question. The process of appearance of recombination nodule occur in which substage of prophase 1 in Pectide? Guys, think about it. What they are asking, that during the what we call cell division time, during the meiosis time, you know that the crossing over will happen, synapses will happen, and the chromosome, they are going to be overlapping to each other, and there will happen exchange of the segments. But at this point of time, a chiasma is present, right? A chiasma is the point where they are attached, and a set of enzymes are present at this point, which include ligase enzyme. We call it as, they create the gap. Yeah, there can be a polymerase enzyme, there can be restriction enzyme. But point is that this is known as recombination nodule. Recombination nodule, where the exchange is going on. When does that happen? In crossing over, I'm so sorry, in meiosis, crossing over happened, right? And that happened in meiosis 1. If we talk about meiosis 1, it includes prophase 1, metaphase 1, anaphase 1, telophase 1, right? And if we talk about prophase 1, it further have 4 to 5 stages. You remember pectitin, diplotin, zygotin, and dikinesis, all these stages. Now think about when it happened. I give you time for that. I think I already answered it indirectly, but that's pectitin, okay? Let me show you a picture, just a second. Okay, guys, so look at this. Into this picture, you people are watching liptotin, zygotin, pakitin, diplotin, all the stages. And you clearly see that into the leptotin, it appears like a bouquet, right? You remember you have learned that how the chromosomes are aligned. And into the zygotin, synaptonymal complex is there, but in pakitin, proper tetrad is present, crossing over is happening. Okay, so that's the thing. So let's move forward. So it's talking about the crossing over. Let's talk further and let's talk about the next question. You already saw the answer, but that's okay. Which of the following stage of meiosis involve the division of centromere? First of all, chromosome has been, what do we call, a chromosome pair is there. And after that, the chromosome pair is getting segregated like this. And during this time, centromere division happens, right? So technically it is asking about when 2n is getting converted into n, right? Come on, tell it. No, it's not talking about the 2n is to 10. And actually, it is talking about how centromeres are getting segregated. You people already are watching. The answer is n of phase 2. But think about it, how it happened. So guys, let me tell you this reminder. During the cells division time, it's PMAT, prophase, metaphase, anaphase, and telophase. Into prophase thing, everything is disappearing. Chromosomes are getting hardened up. They are getting so distinct. During the metaphase time, they all come at the center. We call it as metaphase plate. But during anaphase, they start separating. They start moving apart from each other toward the poles, right? That's when they're starting. So definitely this anaphase is the answer. Anaphase 2. So they have not given any other anaphase into this, right? Even if it would be anaphase 1, that also involves that, right? Chromosomes, they are getting separated. So anaphase 2 is the answer, because though into the meiosis, 2n will become n in meiosis 1, then n become all that equational division happen. But definitely we're talking about the centromere. We're not talking about the mass of the chromosome, right? So you should understand that thing, and its answer is anaphase 2. Let's talk further. Now, guys, next question. During the process of purification, so basically this question is so easy. You remember, guys, if you were there with me in the session, we extracted DNA, and you people have done it in the school also, from the banana, from strawberry, that you were mashing it up and then you were filtering it up, putting it into that dishwasher, putting it with the salt, and then you ultimately add alcohol to it, right? That's when the DNA is extracted. Even a 7th or 8th class student can answer this question. So definitely it is helping with DNA extraction, chilled alcohol. Next question. Family Fabaceae, Solanaceae, and Liliaceae. So when you people were learning it, this in the morphology chapter, this is remember to be like F, Fabaceae first, Solanaceae second, and Liliaceae third. And I told you tips and tricks also to learn it. With respect to the stamen, so we are talking about how these are different with respect to stamen. Pick the characteristic. They're talking about the stamen, right? Stamen is the male or female reproductive part. Many people still don't get confused in it. So that's the male reproductive part. Anyways, so Fabaceae, but not found in the Solanaceae. Guys, before I teach you this option, look at this table. I uploaded this table for the comparative analysis. Into the Fabaceae, you remember Fabaceae is the first, very intelligent. And it is like all 555. I gave you the trick, Mugli Hutti 555, right? It's all 555. 555. Its gynoecium is 1, while Solanaceae, it's like, I gave you the trick, 5 plus 5 is equal to 10, right? So, wait. Into the first one, 5 is equal to 5. I told you this in Fabaceae is equal to 10. You remember that Kg, what do we call? Wait, floral formula is wrong. KGAG, KGAC. Why would they write? It should be KC. Kali Charan Agaya. It should be KCAG. Sequence is wrong in this one. There's a typing mistake. That's PAG. This is also C. That's not G. They have done a typing mistake. It made me confused. Anyways, so KCAG. Now it is right. And this is equal to 5, 5, because everything is 5. And Liliaceae is something which is like teen pagal, 3 idiots. So we were talking about it because we know it's PAG from the pagal. So teen, everything is 3. Now, you do remember, they have again, there is a problem in the floral formula. Guys, I told you that Solanaceae is something which were doing a hard FN and something and it became the CA, while Liliaceae was third and it became the PA. So PA and CA, remember? Epiphilus, Epiphilus, epitapilus, same thing. Yeah. So they have mentioned it, right? Epiphilus, Dithecus. But here they are talking about something is there in the Fabaceae, but not in the Solanaceae and Liliaceae. But that Epiphilus wala thing, it is present in Liliaceae. So Epipetalus also. So these options are definitely out, right? Epitapilus means that the androecium is attached with tepals, right? Petals, I'm so sorry. And Epiphilus means that PA. It is also known as Episepalus or Epitapilus. Wait, not Sepalus. Epitapilus or Epiphilus, because that is where the perianth is attached with androecium. So definitely that is the answer, and this is also. So these options are out. Now let's talk about monoadelphus and monothecus. Monothecus means, theca means one chamber, that into this one chamber is there, and monoadelphus means one pollen tube is found. Fabaceae doesn't happen. It's definitely diadelphus and dithecus. It's a typical one, right? So dithecus means two pollen chambers are there, and diadelphus means two pollen tube will be found, right? Due to that thing. So definitely fourth is the answer. Achha, by the way, let's see if they have mentioned in this picture, carpels, ovary, oval, presentation style, bend, single, simple. Fabaceae bend, single, simple, but united. Not mentioned in the table, but that's okay. You learn it, right? Now you can remember it. That Fabaceae, they are diadelphus and dithecus. They're very simple, not fused. Fused means that's not syncondition. There's no adhesion or cohesion, that kind of thing. So let's talk about the next question. Large, colorful bracts. I don't have to talk about it. Insects are attracted, right? Because of the nectars. So add this insect pollinated question. Spraying of which of the following phytohormones of juvenile conifer help in hastening the maturity of the period that leads to early seed production? Guys, zeatin, this is a type of cytokinin. When it was invented for the first time, cytokinin was the one that they discovered, which helps in meristematic tissue, cell division, growth. Gibberellic acid, it was discovered by Japanese scientists when they were talking about the rice infection, the length because of the Gibberella fungus, it was increasing like too much. But that was the discovery. And after that, a lot of research happened that it helped in increasing the length, it helped in, even if there is genetic dwarfism, it helped us to overcome it. So spraying of which of the following phytohormones not related to the length, but it helped with the juvenile conifers because they are going to increase in size, seed maturation will happen, production will happen, and they will have that length increase and growth. So definitely gibberellic acid, because abscisic acid, they are negative, matlab they are growth inhibitor. IA is auxin, and that is like a rooting hormone, right? So no. So you do understand that, right? Let's talk about the next. Axile placentation. Guys, I uploaded this picture already. Just a reminder. Axile placentation may invagination happen from the outer wall. And that create locules or chamber present inside it. And you clearly know that tomato has that. Lemon has that. Right? But not all of it has that. So look for the answer. They have given it China rose, petunia, and lemon, but you do understand it, right? If we talk about this beans, that is, beans are like on the one side. What do we call it? One side pe hoti hai. Acha, marginal. P also have that marginal. So B option is also out. A is also out. Mustard also have the marginal one. Cucumber, primrose, no. So definitely 3 is the answer. All of the options are like this only. So China rose, petunia, lemon, it's given in the NCERT. So they are the direct answers. Now, guys, next question. Among the eukaryote, replication of DNA take place in, I think, a simple student who is in the 9th class with me, because I've been covering this topic in them in the cell chapter only. So they can help with it, that when the DNA replication happens. So synthesis phase, right? Types of interface. No discussion on this question. Let's talk about the next. How many ATP are needed in the Calvin cycle? I have taught this in my classes. Just a reminder, guys, let's take a look at this table for the Calvin cycle. Look at this table. Now you do know that into this 12, let's write it here, ATP plus NADPH plus FADH. I don't see any FADH in it. So let's write it 0. Let's talk about ATP. Look at this table. Let me use some other highlighter. I hope my OneNote is not confused. Yeah. Look, 12 NAD. Sorry, 12 ATP getting utilized. Here 6 ATP getting utilized. So that makes total 18 ATP, right? Look at the NADPH now. 12 NADPH. So if you want to calculate it, it will be 12 multiplied with 3, 36. So if we add 36 to it, that make total 54. Total 54, you know that 1 NADH is equal to 380. Total 54 ATP. But in this question, they're not asking, they're simply asking how many ATP and NADH. Very easy question, guys. Directly we have learned this. Next is in gene con method. It's used to, I was so sad. One of the students shared this question paper with me and he clicked on some other option, not on tungsten and gold, because it's very easy. I mentioned it in the class also that they are neutral kind of. They don't interfere with the DNA activity. They are inert kind of to them. So second is the answer. The thickness of ozone in a column. This is clearly mentioned in the. I'm so sorry. I wanted to click on Dobson. It is clearly mentioned in the 10th class books also that the ozone, it is measured in Dobson unit. Guys, if you look at this diagram that you saw already, a normal global average ozone length is 3 millimeter, 300 Dobson. Look at this coin. And if you see it on the right side, that's when ozone have a whole average 1 millimeter, which is actually the thinning of the ozone. Okay, next question now. Unequivocal proof that DNA is the genetic material was first posed by? No. Frederick Griffith. Okay, first of all, Hershey and Chase, Avery, McLeod and McCarthy, Wilkinson, Franklin. First of all, you do know that all of these people have done their great contribution for DNA, right? So think about what is the answer. If we talk about Hershey and Chase, yes, they have done experiment. They were, using phosphorescent sulfur radioactive material to prove that whether this is the DNA which is getting in or the protein. And then Everett, McLeod, and McCarthy have done their own experimentation on after that transformation experiment. They were talking about whether carbohydrate, protein, lipid, using all that enzymes. Wilkins and Franklin, they were talking about the DNA 3D structure initially. They did not give much detail. Which is like X-ray wala tha na, crystallography. Then Frederick Griffith, he has done the experiment, Griffith experiment. Everyone know about it, right? I don't have to explain Griffith experiment. You do remember that. If you don't, go into your books. So clearly first experiment was done by Chase. Hershey and Chase. Remember it. Because it is equivocal proof that DNA is the genetic material, because all of them did their work somewhere. They were getting confused between protein, carbohydrates and all that. Wilkin Franklin never was ever, related to these experiments. Next question. In this equation, gross primary productivity minus our respiration is equal to net primary productivity. So here, I did answer the question. I did answer the question. I thought like, I'm actually reading this question for the first time. So that's respiratory loss. Because it's not RQ. RQ is respiratory quotient. It's not reproductive. So that definitely second is the answer. What is the role of DNA polymerase third in the process of transcription in eukaryote? I uploaded this table here. Just a reminder that DNA polymerase is of many types here in eukaryotes. RNA polymerase, RNA polymerase 1, 2, 3, 4, 5, and they are making different components. Like pole 1 is coding for larger ribosomal RNA, pole 2 is coding for messenger RNA, SNO RNA, some snRNA, mitochondrial RNA, and polymerase third transfer RNA, small ribosomal RNA, some small nuclear RNA, and mitochondrial RNA. And this is the again small small interfering RNA, siRNA. Yeah, I'll confirm that. Anyways, guys, now if you look at this question, I don't think this whole explanation in your syllabus, but you should remember this one. That RNA polymerase third here. Reminder what it is coding for. Now let's take a look at the option. Transcription of transfer RNA, 5S RNA, snRNA, transcription. Okay, we do another. Option is first. Transfer RNA, 5S small, ribosomal RNA and small nuclear RNA. So you should know this thing. I've taught this thing in my class. Now let's do the next question. Which micronutrient is required for splitting? That's no discussion. Manganese, because before that, if there is an absence of it, guys, the question is from mineral nutrition chapter, and there is a clear-cut mention on that, and you should remember it. So yeah, it's important. Otherwise, photosynthesis will be affected. In angiosperm, the haploid, diploid, and triploid weight. Let me show you this question. I've recently given this homework to my student to draw the diagram of ovule and show this all kind of structures, whether it is ploid, diploid, or triploid. Chalo, wait a second. I would like to paste it here. So just a reminder, into the ovule, different area are there, and all the area have its own ploidy. Chalazal, nucellus, 2N, antipodal N, synergid cell N, polar nuclei N plus N. Polar nuclei is not diploid, it's N plus N. Integument 2N. Now take a look at the question. In angiosperm, haploid, diploid, and triploid structure. So triploid structure, so obviously you know that when polar nucleus, which is even N plus N, it is going to fertilize with one of the sperm. They all together form 3N, which will be forming PN, primary endosperm nucleus, and then it will be converted into endosperm. So primary endosperm is mentioned here and here. Rest. Haploid and diploid. Antipodal and synergid. Synergid and zygote. Zygote is diploid, definitely. Synergid is haploid. I just talked about it. So yeah, second is the answer. Let's talk about the next question. Next question is the phenomena of pleiotropism refer. Guys, this question you have done in your genetics. I have taught you with the example in your syllabus. So let's read the question. Presence of 2 allele, each of the 2 genes controlling a single trait. A single gene affecting multiple, that's multiple alleles. Anyways, more than 2, wait. A single gene affecting multiple phenotypes, that can be related to it. More than 2 genes affecting a single trait, not possible. Presence of several multiple alleles of a single gene controlling a single crossover, that's totally wrong. So pleiotropism may, though you have seen the answer, I didn't want to show you the answer, but a single gene affecting multiple phenotypic expression, that's a pleiotropism. Just a second, I think I have taught this in the genetics. Wait a second. Actually, I'm looking for that example that I told you. Achha, I have given you example of phenylketonuria example I have given you. Take it. I have taught you guys this one, definitely. So basically into this now, I was telling that how one gene is affecting multiple effects. I gave you example of the pea plant also, that how one gene is controlling height in the pea plant, 3 characteristics. Seed color, seed shape, similar thing. Then I gave you another example that single gene is, controlling multiple trait. Then I gave you example of one more, which is like seed ripening wala tha mere khayal se. I think I'm just going to collect that information. Just, stuck my head. So anyways, I gave you the reasons also that like one gene is there and it is affecting if the 1 and 2 multiple characteristic, because they were kind of crossing that information. The crossover is different, but they were kind of, anyways, let's move ahead. I'm going to just confirm it. Now let's talk about the next given below are this assertion reason. ATP is used at 2 steps in glycolysis. It is actually correct. You remember 2 ATP are formed, 4 ATP are formed, 2 is utilized, so that makes all over 2. Plus then NADPH will also come. So yeah, definitely the statement is correct. A first ATP is used in converting glucose into glucose. Okay. I don't know whether you remember or not, but it is actually correct. 2 ATP are getting utilized. 4 ATP are getting utilized because glucose molecule is getting broken down. And then 2 ATP at one place, 2 ATP at one place. It's like that. So first ATP is utilized in converting glucose to glucose 6-phosphate, where hexokinase enzyme is participating. And second ATP is used in conversion of fructose 1,6-phosphate to fructose 1,6-bisphosphate. That is also where the kinase enzyme is involved. And that's actually phosphofructokinase, PFK, which is the allosteric enzyme. And that's also where the ATP is needed. So definitely this both things are correct, but let's see the option. Obviously, ATP is used at two-step reaction glycolysis, because first ATP that doesn't make a difference. So guys, with this, I think we are done with all the question, and you did understand all the question. I didn't find any question very difficult in your paper till now. I was sitting so relaxed, like before taking the session, I was like, okay, the session is going to be too easy, because then my 9th and 10th class student also can solve a lot of questions. So further question discussion we will carry on in the next class. Share this video with your friends, and you should be watching all the solutions of the NEET exam, right? And further from 126 question will continue in the next webinar. Till then, thank you so much. Bye bye. Take care. Thank you guys. Stay connected.